Welcome everybody to our tutorial to our new software Omnisphero, which was designed to analyze classical endpoints like neural quantification, neural morphology, but also neurosphere-specific endpoints like radial migration and neural density distributions. In this tutorial, we will give you a brief overview on how to work with Omnisphero. So it starts with installing the Omnisphero X file, which will install the MATLAB runtime. After starting the program, you have to load the pre-constructed save. This will give you a well list. Then you have to load the corresponding image folder. And now we can visualize either the nucleus or the neuron channel or a combination of both. Then you can zoom in the image for magnifications, or you can click in our predefined filter set. And you can also display nucleus coordinates, neural coordinates obtained by the Salomics software, and neural coordinates obtained by manual quantification. In the next step, you are asked to optimize the parameters for the automated neural quantification. And after performing the algorithm, you can see the results in neural tracing, and you can display the results alone. Or to display false positives, you can subtract the manual ground truth validation, which results in objects which are falsely identified by the algorithm. Or you can go the other way around and show only coordinates which are not identified, so false negatives. And by zooming, you can see the corresponding false identified or not identified objects. In the next step, we can execute the neural tracing algorithm on all wells at the same time, only applying the filters for calculation time. Or if we have more time, we can calculate the entire well. And again, we can show false positives, false negatives, and only identification by our algorithms. Finally, we can export the well statistics in CSV files, which then gives us the number of nuclei, the number of identified neurons, and from different methods. Furthermore, we can export the analysis of neural morphology. Therefore, you have to install the PostgreSQL software on your computer, which you can find in the link of our description. And after downloading this, you can calculate the neural morphology. You enter the name of the experiment, and consequently, you have to enter your host name and your password. And then the calculation of the neural morphology starts. You can actually follow the calculation by seeing the exported PNGs for the skeletonization algorithm, where we can see in purple and red the cell nuclei and skeleton in blue. And after a while, you will have one skeleton for each identified neuron. Then you can load the library into PostgreSQL, and you can export the morphological characteristics like neurite lengths and so on. And so we can see here the mean neurite length, the total neurite length, and the average neurite length for the 3 wells. Furthermore, we can assess neurosphere-specific endpoints like radial migration, which we can see here by show rings, where we can see the inner ring determining the edges of the neurosphere core, and the outer rim for the furthest migrated cells. Furthermore, we can remove the core to avoid areas with too high cell densities. And again, we can export the migration distances for all wells separately, and also the neural density distributions within the rings of the migration area.